So now we're going to talk about translation. Translation has a lot of stuff going on, and we're going to start by talking about the structure of a ribosome. So a ribosome has two subunits. It has a large subunit and a small subunit. And the large subunit has particular sites on it, and they're known as the APE sites. A is for amino acid, and that's where you would initially have a new transfer RNA bring in an amino acid. P is for polypeptide. This is where you're going to have a growing chain of amino acids. E is for empty or exit, and this is where the empty transfer RNAs are going to exit the ribosome. On the small subunit, this is where the messenger RNA is actually going to bind. And the only time that the large subunit and the small subunit come together to make a full ribosome is when you have messenger RNA present. So we have our messenger RNA strand, which is really just a sequence of bases. And as our messenger RNA binds to the ribosome, we kind of initiate the process of translation. Translation is the process of reading the messenger RNA and incorporating a sequence of amino acids to produce a polypeptide. Polypeptide is really just talking about a protein or a chain of amino acids that can be folded into a protein. So, this is going to be kind of complicated to keep track of, but I'll do my best to keep copying and adding images here. So, we have this strand that our ribosome is going to try to read now. We have codons, and codons are a series of 3 bases on messenger RNA that code for an amino acid. So we read our strand of our mRNA in series of 3. Every 3 bases codes for an amino acid. The next 3 codes for a new amino acid. The next 3 codes for a new amino acid, and so forth. But we don't start reading until we hit a start codon. So a start codon is AUG. That's the official start of a protein. So if we look at this strand that I've drawn here, we have G and C, and we probably have like thousands of other bases ahead of this. And eventually you see AUG. Once AUG is aligned under the A site, we start translation. Now we have these transfer RNAs that come into the A site. And a transfer RNA is going to carry an amino acid. The transfer RNA has a very specific anticodon, and the anticodon is a series of 3 bases on tRNA that bind to the codon of mRNA. So as we have AUG, our anticodon would be UAC. Once the correct transfer RNA that has UAC brings in an amino acid to the A site, we have officially initiated translation. So the first part, messenger RNA, is going to bind to the small subunit of the ribosome. The large subunit is then going to bind to the small subunit. The ribosome is going to move along the messenger RNA until it reaches a start codon, and the start codon is always AUG. Once we've reached the start codon, transfer RNAs are going to attempt to bind to AUG. So numerous ones are going to come in and try to bind, but the only one that can successfully bind is going to be the one with the anticodon UAC, because it's complementary to the codon. It's going to bring in the first amino acid, which begins translation. So now that we have our first tRNA in, the ribosome is going to do a shift. So the ribosome shifts down this way. The next 3 bases are going to come into the A site. The AUG is going to move over to the P site. And the GC is going to go move over to the E site. So now our transfer RNA is here with that amino acid and the anticodon UAC. And now we have a new codon underneath the A site of the ribosome. So a new transfer RNA is going to come in. And again, many are going to try to bind. But the only one that can bind is the one that has the correct anticodon, GCA. The ribosome is going to shift again. So we're going to bring the next 3 bases, the next codon under the A site. This one goes here. This one goes to the empty site. And you'll notice that the amino acid from the first transfer RNA is going to be transferred over to the growing polypeptide chain. And this polypeptide chain is going to stay here and keep adding amino acids. So we have a new transfer RNA come in, and it has to have the correct anticodon. And it carries a new amino acid. And this is going to keep shifting down, reading the messenger RNA in series of 3 bases until we have the termination, or the stop codon, reached. The stop codon is a series of 3 bases that stops translation. And it's on mRNA. So once we hit that stop codon, then we stop translation. I've posted some videos on the Blackboard site about transcription, translation, DNA replication, so I strongly encourage you to look at those videos. The videos give you real animations that are pretty good at kind of following along with the process here, but hopefully this makes some sense to you guys.